Yeah.
Oh, yeah.
I don't know.
She takes her to the HR so much.
Yeah, because a lot of people are already like.
She won't be able to.
Yeah, pretty much.
I will be. I will. Someone.
Right.
My dad's in the
Yeah.
ER.
Yeah.
Do you think it helps us with conversations?
No, it's not for great.
Not for.
Is that just? Yeah, we need to not.
Yeah.
It was really hard.
She can also, especially
Just
with my
go
dad
random.
when dimensions, because like we tried to sleep and me and my mom would be loud, so I just break it. Like
Yeah. Yes, and then
we could never figure out.
Yeah.
They're fine, it's 7 a.m.
All right, guys, so this weekend I finished going over the midterms. There was a bit of a mix of results. So I was looking at the class distribution, trying to see what's going on. There were some areas where we had some like really like weak performances, which I'm like, hey guys, the midterms are a little bit hard. Like for me, it was like I used to love to cram. And this class is really hard to cram. So it's like you kind of have to study beforehand, right? The other one is, so when I was looking at this, it's basically like, in this part is like how we were doing with that first section, vocabulary, right? So that's like the distribution of what I got in that section. And like as a class, we did like 68%, right? So it's like a lot of you guys lost a lot of points with just the vocab, right? So I'm a little bit worried there. I think in the class, I was trying to emphasize it a lot when I was like, hey guys, remember the RNAs, there's a bunch of them, they have their own little code for which one you're working with. But I feel a lot of you guys got tricked with those questions, with the RNA questions. So that kind of brought the score down a lot in the beginning. From there, so if everything looks red, it's a good performance. When I see green and yellows, it means that like your yellow is 70% of the grade, green means you're below like 50%, right? So in some sections, you guys did very well, like the multiple choice things, you guys were fire on that. That was really good. And these two are the last two free responses. You guys gave me a lot better performance on the second one than you did the first one, right? So I'm like, maybe the first one is because it was like the first material and it was like over that Zoom call. So I'm like, I hate teaching over Zoom. I love to be able to look at you guys' faces, be like, if you're nodding, you're falling asleep, you're just kind of like, I don't know what's going on. It helps me. So I'm like, maybe it was that. But what I'm thinking is there is a regrade, right? So, sorry, I dropped one of the midterms. This are the tests. This is one. We have 3 more to go, and I'm gonna drop the lowest score. So if you don't want to do anything about it, I'm okay with it. We'll just keep going, right? Just make sure that you kind of think about what's going on in the class. Like if you didn't have enough time to study, or if you're like, hey, I didn't, set up myself enough to win at this test, make sure that you ramp yourself up for the next one, right? To be like, hey, I'm going to be more organized or find a better way to pick up the material for myself. If you're having trouble, reach out to me. This is what I love to learn, how to learn. So we can go talk about different techniques of ways for you to improve your notes or different ways for you to study your notes. If you're having trouble, reach out to me. If you kind of let off, if you didn't go study too hard on this one, then make sure you step it up for the next one, yeah? I want to see you guys do well. And it's just the 2 crews. If you did well, good job, appreciate it, thank you. It was good reading your responses. Throughout everyone's test, I saw that they had really good responses. And then I'm not really good at giving that much positive feedback, so if you don't hear like too many good notes on my stuff, I'm sorry, but I don't do it too much. But you guys do have creative stuff. You guys show me some areas like you guys like really understand this topics, and you guys did a good job. So like you guys are like in my book, you guys are really good. Like you guys are doing good. But we'll do better on the next one. But with this one, like I said, if you want to let the score go, it's fine. We'll be like, hey, this is the one you drop. If you want, what I'm thinking is for the definitions, the first 2 questions and that first multiple choice question, or sorry, the first free response question. I'm going to give you guys the midterms back. If you guys want to redo those questions and I'll give you your average score, right? So it's like, I'll give you the test, take this whole week, next Monday, give me your midterm back, and it's like, I want you using all the resources that you have. I want you to emphasize using your notes, the class lectures, and in any of the videos that we have for the class, go through that and make sure that you feel comfortable, like, hey, I was able to clearly find the material where it was at, how it was presented to make the storyline, right? So don't try to Google it or find the answer another way. Go back to our class material. That way, maybe you can see how I place things, and then better ways for you to pull them out or try to identify them. Yeah, so if you do that way, again, I'll hand them out, give me the feedback, or give me the test back by next Monday, and then I'll update your grades for that section. Yeah. So, yes, sir.
So you're saying like giving us the exam back, do you just want us to like, on the ones that we got wrong, do you just want us to write like our alternate answer off to the side? Or do you want us to write?
Get a brand new piece of paper.
Okay.
Because I'm thinking for this first part, the definition one, this is one of the hard ones. There was an answer you put in, and there's the correct answer. Right? So then tell me what the correct answer is, and then tell me why the other one is not. Like, what is the other thing belongs to, right? So, for example, like if we're talking about the small RNAs, right, which are normally with the spliceosomes, like that would be one of your answers. Whatever you answer you put in that spot, you put another RNA, tell me what that RNA belongs to. And you can be like, hey, this is the one that belongs to this. This other RNA belongs to a different system.
And you said it's for the matching section, the first two questions, and the first free response?
Yeah.
Okay.
Those are the big ones. Everything else, the other ones like the class, in my book, I was almost looking like anywhere where the class performed below 70%, made me think that like, if it's a class effect, and that's most likely it's me doing a bigger effect to you guys, right? If you guys as a class score normally higher than 70%, it might be like, oh, it's one or two people who might have, missed it, but everyone else got it pretty well. Yeah. Yeah, so that should be good. Yeah, I was trying to figure out, guys, I graphed this so many different ways. I was trying to figure out how the thing goes. So this is like the class distribution. So you can see this is the grade levels. But then I saw the vocab brought us down a lot. Like, we scored really low. So that's the one I was like, let me target that one. Yeah, the distribution was a little bit shifted down. So vocab, guys. But you have your midterms back. Go over them. If you have any questions, or if you want to argue and answer with me, I come do it. I enjoy doing that, because it'll be like, there's a few of them there, which I know I marked them wrong, but I'm like, I think you can argue them. Yeah, so it's like, it's up to you if you find them. They might be in other sections, like in the multiple choice, I messed up and I put 2 correct answers on part C. So 2 of them are correct. So I'm like, for those, I gave you, like if you picked either one, I gave you the points. But there's other spots where if you argue them, you could potentially get away with it. Like you're not get away with it, but it's like technically you are right if you argue. So read through them, and I will do regrade and stuff, so that should be fine. We'll pick them up, I'll give them at the end of class. So they should be pretty quick. The topic is CRISPR, so it's pretty, once you see the mechanism, you're gonna be like, awesome, this tool. Yeah. Any other questions? No? All right. Yeah, no pressure to redo the regrading for the midterm. Again, if you're like, hey, I'll do better on the next one, I'm okay with it too. I just wanna make sure I keep it open for you guys. Where are we? This is mine. Oh, by the way, Anne, thank you very much. Appreciate it. Your question, your favorite pathway.
Yeah.
Half the people in the class wrote about it.
Oh, yeah.
So, you know what I mean? I was like, good discussion, dude. Yeah, you guys kicked butt on that one. Good job. And who was the other person? There was only one person who picked the ATF6. And I was like, that was the easiest pathway. And I'm like, nice, good job. And then the rest did the IRE, so that was good. I was happy. All right, so let's talk about today. So today we're kind of talking about how we can mess with the genome or how we can modify it, right? So for this, we're gonna be talking about how is it that you can change it? And to be honest, there's gonna be different ways that we're gonna try to grab that DNA that we care about and introduce some kind of mutation to it, right? And when you do that, we're gonna be relying on the repair mechanism. So this is gonna be the two that we talked about last week. It's HR and NHPJ. So again, those two definitions, for sure they're gonna be on the next midterm. I'm gonna write in the letters like that, and you can tell me exactly what they mean. Right? So for this, HR, homologous recombination, non-homologous engine. Well, we'll talk about how these two different techniques of the DNA getting repaired, they're slightly different from each other, and despite those differences, basically give us a really powerful tool set that we can do. All right, let's get started. So let's look at them. So on the left, you have your homologous recombination, right? So in this case, I want you guys to be thinking about, again, you have two different copies of your chromosomes. In my head, I always think, I got one from my mom, I got one from my dad, right? In this case, it said my dad's the light brown version. When that one, a double-stranded break happens, you basically lost connection to those two molecules. In this case, there's special proteins that come in. There are nucleases that eat away at your DNA, and you normally need a 3' overhang. You do something like this. So you eat away 3', keeping that gap, and you do the same on the other strand. This is really cool because those two single-stranded pieces of DNA, they basically serve almost like guides that help you go find a partner onto the chromosomes of your other parent. So again, if my dad's copy broke, I'm gonna go look into my mom's copy of that gene. That single-stranded part helps me find the perfect target. So when you're matching up, and at that point, this other strand, the other strands basically match up into each other. This matching up then lets us bring in a polymerase right here, and we can start copying 5 to 3 prime. We'll start filling in this gap, giving the base pairs. Because we do this, this part helps us find the right target, and here means that whatever we broke off, we can get the target matching to that sequence, right? Eventually the ligase comes in, we glue it back together, and we can get our DNA back out. My dad's good copies, where my gene broke, I'm gonna take a copy of this from my mom's gene to bring it up to be able to replace that sequence. The cool thing when you're done with this is because you're using a guide to help fix you, if when after the break, there's a chance that there's DNA molecules that can come and add stuff to your DNA, or there's stuff that can eat away at the DNA. If that normally happens, this thing doesn't care. You can take off 100 base pairs. All that's gonna happen is we're gonna match up to a further apart spot, right? We'll match up here, and it still fits everything that we're missing. So to make sure that our code is perfect. Big thing here. On this side, this is the brute force attack, our brute tough way freeze. What this one's doing is we have the same double-stranded break. At this point, we have this cool heteroranders. What they're doing is they're recognizing these open-ended, these two blunt ends of DNA. They bind to them, they basically bring them together, and you get this ligation event to happen. You just glue the two things together. The problem is, after your DNA break, if you lost 1, 2, or 3 bases, this thing has no way of knowing if something got bigger or shorter, or like caused an indel, right? Insertion, deletion. It's literally just gonna grab the two and just pop them together and be like, this goes next to each other. Right? So this kind of fixing of the DNA is error prone. So here, a huge chance of being errors. Where we're super sensitive with this is if this happens, where we have one of our genes, specifically the coding sequence. Right? So remember, a coding sequence is set up like perfectly spaced 3, 3, 3, 3 base pairs to give us one protein. If you mess up, add one base, take away a base, you're basically going to shift all the amino acids after that and ruin your protein. So super dangerous, right? The other one is going to be really good because it's going to be able to fix things. So now, how can we take advantage of this, right? So the way that we're going to do that is, back in the day, the old way to try to figure out how genes work or how things function is, we still kind of do it, but we would get like a really dangerous chemical. I would get like, maybe like 10,000 seeds. I wash them with this really harsh DNA chemical, do all this DNA damage. I'd grow a bunch of seeds, and then the goal was I would search for a phenotype, hoping that one of those plants would have a mutation in the gene that I cared about. But I'd be screening thousands and thousands of plants, hurting them, trying to figure out where my gene was. Because my damage before was just randomly landing on the chromosomes until I hit the gene that I want. This new tech that they've been developing is, how do we go from, instead of trying to damage the genome at random to find what we care about, to how do we mess up exactly one gene or one little piece that we care about? So these top techniques up here, these meganucleases, zinc finger nucleases, and these tau effect nucleases. They were like the initial step of how we figured this thing out. And it was, for the most part, you're gonna see them combining a protein that likes to bind specific sequences of DNA, and then they're combining it, like literally doing two proteins, trying to put the two RNAs together so that you can make two proteins from a single polypeptide that would have an endonuclease. So it can go grab the DNA that it cared about and then cleave it at that spot. Right? So that was the old school way. Then down here, a little bit at the end, I'm talking about the newest one, which is CRISPR. This one, instead of having a protein find the target and then the endonuclease, this one actually uses a piece of RNA. The RNA gets us right to the DNA we care about, and then the endonuclease will cut it. So now we're zooming into these three different techniques. So we have here first that meganuclease, right? In this case, it's going to work as a dimer, but it's going to be having these two different sequences that it's going to be using to find its target, right? Once it finds those sequences, then it can do the endonuclease function and it cuts your DNA. The zinc fingers, these are basically, these would be the little zinc structures. You glue 3 of them. Based on what pattern you put here, it'll tell you what sequence you can try to grab onto. And then this is your FOK1, is the endonuclease. This is the one that basically do the double-stranded break on your DNA. Just like that, another system that we're developing was talons. But again, this is proteins that they're building up that bind to the DNA. You modify these to go find a specific sequence, and then you bring in your endonuclease to just at those sides. So the goal here is try to find a specific place to mutate. You have to work with these pretty it's pretty rough trying to get the protein to perfectly match. If you want to target a new gene, you have to go back and remodify this protein, which in the lab is kind of tough, right? Let me see. But the goal from, so let's say we were developing this in the lab back in the 90s. We made this, we would introduce that first break. The DNA just split open on you, and it's like there's different things that could potentially happen at that site. Right? So when I was talking about that, if this is, you imagine, this thing would represent almost your 2 chromosomes, and this is your 2 chromosomes, they were initially joined together. You can do this, like again, with radiation, endonucleases, or other chemicals, but once the double-stranded break happens, how are you going to repair it? If you did the non-homologous end joining, just glue stuff back together, right? If DNA got deleted, you have an issue with deletions, right? Piece of DNA that will be missing when you put it back in together. If stuff gets inserted or copied back in, again, non-homologous end joining doesn't care. It's going to grab those 2 pieces of DNA, pop them together. In some cases, when you have the breaks, there's these inversion events where, like, it'll be like a cable, like this. And like a part of this almost acts like our lyrians. Like it'll be like, this thing will break off from like here. This thing goes, and it actually binds here first. Then this one comes back and binds on this side. So it does like a little intermediate thing where it kind of swings out. Like it makes an extra break kind of further upstream of it. But either way, you end up with like this inversion of the genes in that region. You can flip them over. Then let me see. And again, sometimes you get like these duplication events. But I remember with this one, it was more like you had 2 cables like this. And if the double-stranded break happened away from each other, like this, when this thing lets go, it would eventually glue itself back together. But it would do it like this. Like that. So instead of being like, hey, these 2 match up and we need to fix this, it would let them push apart. It would think this is the blunt end, and it would fill this gap, and it would fill this gap. So what ended up happening is this little region here would end up creating 2 different copies. Yeah. So these are all like the messy ways of trying to fix your DNA that end up giving us something that's wrong or mixing it up. And then from this one. So this is mainly looking at non-homologous end joining, right? In this case, it's when you have homologous recombination, right? Which means you can use a different piece of DNA to help fix the one that's broken. Normally in our cells, what's happening is we use, like I said, our dad's chromosome, we use our mom's chromosome to fix it off of it, right? The other way that we can do it now in the lab is we can actually clone a piece of the gene ourselves. What we do is in the purple part, I can put whatever I want, right? This could be like, if they say you're missing the gene, I can put the proper version of the gene here. And all I have to do is make two regions, this red and this orange one. I just make those to be similar to the places surrounding where I'm gonna cause the break, right? So that way, after I cause the break, that piece of DNA is gonna be looking for DNA to try to copy or be similar to get the version off of it. When it's doing that, instead of finding the other chromosome, it's gonna find my DNA. At that point, it's gonna use these two things to build that D ring, right? Kind of like this. When it's trying to build this complex, instead of doing it to another chromosome, my DNA would be right here. This should be that red region, this should be the orange region over here. It clamps onto my piece of my DNA, and I can either put something bigger, I can put something smaller, I can get rid of a big chunk of DNA, but basically it gives me that huge power of putting DNA in and taking DNA out. In this case, they've done some pretty cool stuff where you can, again, if the gene's broken, let's say the mutation, like you inherited a mutation and your gene is not good for you, you could technically go in, target that piece of DNA, cut it. We can put in a good copy of the gene just like this, go to that same site, cut around it, and we can glue in the proper version, or a better version of the gene, and you can fix it. When you're studying different genes, you can also do the same break, but instead of targeting the gene itself, you kind of target it in the back. And you can put in a different protein there. Like if you, like for me, I love to put fluorescent proteins so we can see them in the microscope. You can literally bring in the gene for something that fluoresces, and you can glue it in the back of your gene. And now when it gets made, we'll see your protein being made, and that fluorescent protein, I can look at it in the microscope. So super powerful technique. All right. Okay, so then. All right, I think I'm gonna skip this, guys. All right, now CRISPR is the bigger, newer one. So this is like the super fancy one, and it's super more powerful than the ones we've seen before. In the sense that, again, before you had to build these proteins and put them in the right order to go find your target sequence. If like your lab worked with one gene, you modify these three different proteins to go target the DNA around that site. But if you wanted to mutate it to a different gene, you had to completely redesign those proteins, put them into the experiment, try them out to see if the proteins actually bound the sequence that you wanted to. So it wasn't perfect. The CRISPR-Cas9, this one is super, super powerful. It comes from bacteria, and then so it's a two complexes, right? So we're gonna have an RNA component, and we're also gonna have a protein component that's part of it. The protein is the one that's gonna be the big endonuclease, the big cutter. So the cool thing about this is that RNA part, we kind of know the structure that it likes. So these drawings, they come out like this. They have a little bit. Some people call this the spacer. So this is the sequence that we care about. Then they have like this little stem loop thing, like this, that builds on itself. That looks something like that. Right? So this is your guide RNA. This single-stranded portion, we can basically switch out and put any base pair that you want. Making these in the lab takes like 3-4 days. Like it's fast. We can make this pretty. And the cool thing about it is because they're about 20, like around, what's it, 21, 22? Or let's say it's roughly 22 base pairs. At 22 base pairs, the sequence is almost so perfect, or so it's like, they're so different. You can literally design this to only go target one gene out of all the chromosomes. Right? So out of 100,000 genes that we're working, you can literally go and just break exactly one and not hurt anything else. Super easy to design, super, like, you can switch them in and out, nothing happens. Then the next part to this is gonna be our Cas9, right, which is that green, that's the actual protein, and that's the endonuclease, which is DNA cutter, right? That thing's gonna basically build a complex around this molecule, and it'll serve as, together they'll guide themselves using this to the target gene. So in this case, you can imagine the black here would be like your actual DNA from one of your chromosomes. There's your Cas9, right here, this little spacer sequence, like the one you designed, it matches up perfectly with your target, right? When it's doing that, this protein then does a little cleaving and it cleaves this little thing called the PAM site. I think it's the adjacent motif. Spacer adjacent motif, I forget it off the top of my head right now, but basically, you just use this sequence is to find the target, it uses these base pair, and then boom, you can cut the DNA, introduce your double-stranded break, right? So now, this is super powerful because I can hit anything with this, right? Any gene, any plant, any animal, most model systems, people have done this in already, right? So the cool thing is, now you have this, we introduced the mutation, you can target it. It's super efficient to make, it's efficient to transform, and it keeps going. So as long as, if it finds a sequence that matches this, it's gonna go there, and it's gonna keep cutting, right, until it happens. The cool thing with that is, if it's on our side and we're trying to do an experiment, and the goal is to take away this gene, if, for example, you do the double-stranded break, these two cables become free. If your mechanism, let's say, decides to go down your homologous recombination, right? And it says, hey, there's a double split, and it uses the second copy of the gene to fix itself, it's gonna look exactly the same. It's gonna go back to looking at how it was before it was cut. In which case, CRISPR stays around and it's gonna come back and be like, cut it one more time, right? Only when it goes down the non-homologous end joining, right? If we do the different ways that it can come out, if you split it open and it's, let's say some things start eating it away, right? Or things, base pairs got inserted or some kind of flip happens, then that sequence that we used to target, it's gonna be messed up, and at which point CRISPR doesn't work anymore because it's not the right sequence, right? So this thing literally keeps going, making the mutations until it looks like the way that we want it. So for non-homologous end joining, if you wanna break a gene, that's all you have to worry about. If you wanna go with the homologous recombination, or this guy, right? Normally, when you do that side experiment, it's when you want to bring something in. So if something is broken and you want to fix it, you can break the gene. And again, instead of using the other chromosomes to copy off, what you can do is you can actually put in the exact piece of DNA that you care about, just with the similar flanking ingredients. Does that make sense? Yeah? All right. Cool. So this is that's the main stuff for today. Today's gonna be a really quick day. Everyone feeling comfortable with how to break the DNA? Yes. CRISPR-Cas9 is everyone's favorite. Everyone's cool with that one? Yeah. These two pathways make me a table, right? I want to see you guys talk about how does each pathway work, and then the outcomes of it. Like when it comes to which one creates something that's good, or sorry, which one is more, has higher fidelity, like the stuff stays more likely to stay the same, which one has the lowest fidelity, which means it has the ability for mistakes to show up. And then when it comes to biotechnology, about bringing in pieces of DNA and new ones, know that we're basing it off this pathway, and all I care about is how the flanking is to you up. Yeah, so make your little table, make it look nice. I think for the midterm, I'll probably put a question about how to use one of these texts to play with the gene. Yeah. So either breaking the gene or fixing the gene.
So CRISPR just is a precise break in that region, and if you don't add the new DNA that you want it to be, then it will go through the non-homologous.
So this one happens a lot in our older cells. This one happens more in like at the embryo stages and I think in stem cells. For us, we have a higher chance of this happening, right? So that's unfortunate for us if we do CRISPR on the old cells, we might not get it to fix properly. But people have had luck where they put in really high amounts of the CRISPR protein, like the whole complex, and they put in that, they put in this, that piece of DNA that you really care about, right? And then you have higher chances of that thing trying to recombine it. So experimentally, you have to play with it. Any other questions? No? All right, cool.
I kind of. That's usually like this. 3 hours. And I like the average, it's like.
So I feel like
Our
it's
so.
our original.
Yeah, that was fun.
Average is
And then giving everybody the
No,
same.
it shouldn't be.
Yeah,
It shouldn't be. It shouldn't be.
But.
It'd be very.
As we know how that usually
Maybe I need.
Dr. Jaws is like
That's
apologizing
the thing.
to everyone who has these documents and issues, yeah.
First.
So, yeah, right, first 2 questions. And the first.
I can't even know that anymore. I'm tired of being that.
Okay.
I think it's just intense.
Annie?
Oh, no.
Wait, so but.
Like know the procedure on how
The first, well, so I think it was the one right before talking about.
answers to the parents.
Oh, no, I'm
sorry.
Or the, three of the.
I think I'm locked up right now. I don't know. Mary.
Then of course he said, you can argue any of them.
Yeah, I'm
Hard to yell that. They're gonna take a debate class.
Mary,
Messed it up. Oh, I do
I
this.
saw that.
Ava.
Right?
I don't have to.
That's good.
I think.
I actually, I saw that you said that.
You like.
Did everyone get one? Yeah.
Yeah.